ourselves. There's some basic things that you would want to understand. Two great examples are drugs, Prozac and Wellbutrin. These are commonly prescribed antidepressants. Now remember, we're assaying genes. Genes send the instructions to make proteins. Proteins are targets for drugs. So drugs bind to proteins and either turn them off, etc. So if you want to understand the action of drugs, you want to understand how they're acting in the ways you want them to, and also in the ways you don't want them to, in the side effect profile, etc. You want to see where those genes are turned on. And for the first time, we can actually do that. We can do that in multiple individuals since we've assayed two. So now we can look throughout the brain, we can see this unique fingerprint, and we get confirmation. We get confirmation that indeed the gene is turned on for something like Prozac in serotonergic structures, things that are already known to be affected. But we get to see the whole thing. We also get to see areas that no one has ever looked at before, and we see these genes turned on there. Is this interesting side effects? It could be. One other thing you can do with such a thing is you can, because it's a pattern matching exercise, because there's a unique fingerprint, we can actually scan through the entire genome and find other proteins that show a similar fingerprint. So if you're in drug discovery, for example, you can go through an entire listing of what the genome has on offer to find perhaps better drug targets and optimize. Most of you are probably familiar with genome-wide association studies in the form of people covering in the news saying scientists have recently discovered the gene or genes which affect X. And so these kinds of studies are routinely published by scientists, and they're great. They analyze large populations, they look at their entire genomes, and they try to find hotspots of activity that are linked causally to genes. But what you get out of such an exercise is simply a list of genes. It tells you the what, but it doesn't tell you the where. And so it's very important for those researchers that we've created this resource. Now they can come in and they can start to get clues about activity. They can start to look at common pathways, other things that they simply haven't been able to do before. So I think this audience in particular can understand the importance of individuality. And I think every human, we all have different genetic backgrounds. We all have lived separate lives. But the fact is, our genomes are greater than 99% similar. We're very, very similar at the genetic level. And what we're finding is actually, even at the brain biochemical level, we are quite similar. And so this shows it's not 99%, but it's roughly 90% correspondence at a reasonable cutoff. So everything in the cloud is sort of roughly correlated. And then we find